Moving on in Chapter 7 for Lecture 3, we're going to be discussing pyruvate oxidation and the citric acid cycle. These two processes come immediately after glycolysis in eukaryotic cells, and both of them take place within the mitochondria. But before we get into the details of those two processes, we need to review a couple things about the mitochondria. So you can go back into Chapter 4 and read the specifics about the mitochondria, but I'm just going to point out a few things about the mitochondria anatomy, how this organelle is organized and laid out. Okay, so this organelle has two phospholipid bilayers. It's got two membranes, an outer membrane and an inner membrane. Between the outer and inner membrane is a space called the intermembrane space. Inter meaning between, okay? So between the membranes. The space enclosed by the inner membrane is called the matrix. And the matrix is really important. It's rich in a lot of enzymes and other molecules, and there's just a lot going on inside the matrix. The inner membrane of the mitochondria is very highly folded. And the purpose of that folding is to increase the surface area of the membrane. Well, why would that be important? That's important because the inner membrane is where lots of important transmembrane proteins are located for the electron transport chain and for ATP synthesis. So we're going to talk about that more in the next video. But the more membrane there is, the more of those proteins can be in the membrane, facilitating the reactions that produce ATP. All right, so just to reiterate, we've got the outer membrane of the mitochondria. We've got the inner membrane of the mitochondria, which is very highly folded, okay? Between the two membranes is the intermembrane space. And then the region enclosed by the inner membrane is the matrix. Okay, so out here would be the cytosol. Okay, so we've got the outer membrane, the inner membrane, the intramembrane space, and the matrix. And remember that both of these membranes are phospholipid bilayers. So everything that you learned about membranes applies to the membranes of the mitochondria as well. All right, well, at the end of glycolysis, we were left with pyruvate. And remember, if we're looking at our energy progress curve, we're still about right here, okay? Pyruvate has a lot of energy. Well, pyruvate is going to diffuse from the cytosol into the matrix of the mitochondria. If we're talking about this 3-carbon molecule crossing those membranes, then we're talking about facilitated diffusion. So I'm just using these words to jog your memory about things we talked about earlier in the semester. Okay, well, if pyruvate is oxidized, what does that mean? Oxidation is loss. Loss of what? Loss of electrons, loss of energy. Okay, so pyruvate being oxidized means that something else has to be reduced. Well, what is that something else? NAD+. Okay, so NAD+ is going to pick up electrons from the oxidation of pyruvate to create NADH. Okay, so now we've transferred energy in the form of electrons from pyruvate to NADH. Well, pyruvate is a 3-carbon molecule, okay? Part of the reactions, the oxidation of pyruvate, one of those carbons is cut off. Okay, so we go from a 3-carbon molecule to a 2-carbon molecule. That carbon is lost as CO2. Okay, so CO2 is a waste product of aerobic respiration, and you know that it is what you exhale. Okay, so animals exhale CO2. Other organisms like plants are going to release CO2 when they're doing aerobic respiration. Fungi, protists are going to release CO2. Okay, so we've got this 2-carbon group. 2-carbon groups are called acetyl groups. And the last part of pyruvate oxidation is the addition of this chemical group called coenzyme A, or abbreviated CoA. Okay, so you don't have to stress about what that actually looks like. You just need to know that the 2 carbons that were left from pyruvate have a CoA attached to them. Okay, so in a nutshell, pyruvate diffuses into the mitochondria through a series of reactions. CO2 is released, CoA is added, and we wind up with acetyl CoA, and NAD plus is reduced to NADH. Okay, so the oxidation of pyruvate is an energy harvesting step. That energy is now in the form of NADH. But we just got 1 NADH per pyruvate. Sorry, dog's barking in the background. How many pyruvates did we get per glucose? We got 2, because remember glucose is split in half. And so we wound up with 2 pyruvates per glucose. So each pyruvate is going to be oxidized. So per glucose, we get 2 NADHs as a result of the oxidation of pyruvate. But lots of energy is still stored in acetyl-CoA. Okay, it's still a high energy molecule. Acetyl-CoA is present now in the matrix of the mitochondria. My dogs are having a good time. And acetyl-CoA is going to be the starting substrate of the citric acid cycle. Now, the citric acid cycle has a variety of names. All right, you're going to hear it referred to as the citric acid cycle. I tend to call it the Krebs cycle. It's named the Krebs cycle after Johann Krebs, the man who kind of figured all this biochemistry out. You may also hear it called the TCA cycle. Just a variety of things, okay? But the citric acid cycle, as the name implies, it's a cycle. So it's a series of reactions that ultimately the end product, whatever you start off with up here, you wind up with that at the end of the cycle, okay? The citric acid cycle takes place in the matrix. So there's all of these enzymes present in the matrix of the mitochondria. The most important thing I want you to know about the citric acid cycle is that most of the energy from glucose is harvested here, okay? In the citric acid cycle. This is where most of the energy of glucose comes out. And if we just look at this summary right here, we see NADH, we see ATP, we see FADH2, and we see NADH again. Okay? So lots of energy is coming out most of it as a result of redox reactions that are producing these electron transport molecules, and a little bit of it directly as ATP through substrate level phosphorylation. Notice that we also lose CO2 in the citric acid cycle. Again, waste products that we are exhaling. Okay, so let's look at this. Again, I'm not going to stress about the starting reactants or all of the different intermediates. But the citric acid cycle is going to start with a 4-carbon molecule. Okay, acetyl-CoA is going to come in. Now, where did acetyl-CoA come from? It came from the oxidation of pyruvate. Okay, so we've got a 4-carbon molecule plus our 2-carbon acetyl-CoA, and we get a 6-carbon molecule. Well, if this is a cycle, by the time we get back here, we've got to be back to our original 4-carbon molecule. That's why we lose 2 CO2s, okay? Because we've got to get our original starting product back, okay? Over the course of this cycle, we're starting with acetyl-CoA. And there are going to be several energy harvesting steps, okay, in order to pull the energy that's left in the chemical bonds out, right? At the end of this cycle, we're back to that 4-carbon molecule that we started with, and it's going to be ready to be combined with another incoming acetyl-CoA. Again, let's go back. What do you need to know? Acetyl-CoA comes in. There are 2 acetyl-CoAs per glucose, 1 per pyruvate. So when we talk about going all the way around the Krebs cycle, we call that 1 turn. Okay, so 1 turn of the cycle. Acetyl-CoA turns the cycle 1 time. Well, how many times does glucose turn the cycle? Twice, because we get 2 acetyl-CoAs per glucose, because we had 2 pyruvates per glucose, and each pyruvate was oxidized into acetyl-CoA. Okay, so these products, 2 CO2s, 2 NADHs, ATP, FADH2, NADH, those are all per turn, okay? So per turn of the Krebs cycle, we get 2 CO2s, 3 NADH, 1 FADH2, and 1 ATP. Well, we multiply that by 2, and that's how much we would get per glucose. Now, do you have to memorize all of those numbers? No. What I need you to understand is this right here, okay? If you go back and you look at glycolysis, we got 1 NADH and 2 ATPs per glucose. Oxidation of pyruvate, we got 1 NADH per pyruvate. But in the Krebs cycle, we get 3 NADHs, 1 FADH2, and 1 ATP. Most of the energy in glucose comes out in the Krebs cycle. Okay, that's what I need you to know. This right here. This is the most important thing. Okay, so, but even then, what was the goal of aerobic respiration? To make ATP. Well, we still haven't made much ATP yet. At this point, most of our energy is in the form of these electron transporters. Okay, well, when do we get to ATP? In the next lecture.